As you know, glycolysis and the Krebs cycle together result in the formation of reduced coenzymes such as 10 molecules of NADH plus H plus ions and 2 molecules of FADH2 and 4 molecules of ATP. These reduced coenzymes need to be oxidized to release and utilize the energy stored in them. This is made possible by the transport of protons and electrons from these coenzymes to oxygen through electron carriers present in the inner mitochondrial membrane. This metabolic pathway of electron transport is called the Electron Transport System, or ETS. ETS serves 3 important functions in aerobic respiration. It regenerates the oxidized form of coenzymes to be used in glycolysis and the Krebs cycle. It transports 2H plus and 2 electrons to oxygen, and it utilizes the energy of the coenzymes in the production of ATP. ETS comprises several electron carriers that include NADH dehydrogenase complex, or complex 1, ubiquinone, or complex Q, succinate dehydrogenase complex, or complex 2, cytochrome BC1 complex, or complex 3, cytochrome C, and cytochrome C oxidase, or complex 4. Electrons from NADH plus H plus are transferred through complex 1 to ubiquinone, and protons are moved from the matrix of the mitochondria to the intermembrane space. In the same way, electrons from FADH2 are transferred through complex 2 to ubiquinone, and protons are moved from the matrix of the mitochondria to the intermembrane space. Ubiquinone transfers the electrons to complex 3. Complex 3 transfers the electrons to complex 4 through cytochrome C. Here too, some protons are moved from the matrix of the mitochondria to the intermembrane space. Cytochrome C is a mobile carrier attached to the inner membrane of the mitochondria and helps in the transport of electrons between complex 3 and complex 4. Complex 4 contains cytochrome A and cytochrome A3. It transfers the electrons to the final electron acceptor, oxygen. Oxygen, on receiving the electrons, reacts with 2H plus ions and reduces to water and thereby drives the ETS. This reaction can be summarized as 2H plus 2E minus plus half O2, which results in H2O plus energy. Now let's see how the electron transport chain is coupled to ATP synthesis. As you saw, the electron transport and movement of protons creates a proton gradient across the mitochondrial membrane. The protons in the intermembrane space need to be pumped back into the mitochondrial matrix to maintain equilibrium. As the mitochondrial membrane is impermeable to protons, they are pumped through a membrane protein called ATP synthase, or complex 5. ATP synthase consists of 2 components, F0 and F1. While F0 is an integral protein that is used as a channel for proton transport, F1 is a peripheral protein and a catalytic site responsible for ATP synthesis. The energy derived from the proton pumping is used for the synthesis of ATP. Thus, when two protons are pumped from the intermembrane space to the mitochondrial matrix, one molecule of ATP is synthesized. The number of ATP molecules synthesized through ATP synthase depends on the nature of the electron donor. For example, oxidation of NADH plus H plus results in 3 molecules of ATP, whereas oxidation of FADH2 results in 2 molecules of ATP. Aerobic respiration uses energy conserved in coenzymes during oxidation-reduction reactions to create the proton gradient required for phosphorylation, unlike photophosphorylation, which uses light energy. This is why the aerobic respiration process is also known as oxidative phosphorylation. The net gain of ATP produced during aerobic respiration can be calculated based on the assumptions that the glycolysis, Krebs cycle, and ETS takes place in sequential order. NADH plus H plus produced during glycolysis is transported into mitochondria for oxidation. The intermediates formed are not used for synthesis of other compounds, and only the glucose molecule is respired and no other molecule is used as a substrate for respiration. Based on these assumptions, we can calculate that aerobic respiration results in a net gain of 38 ATP molecules from complete oxidation of the glucose molecule into carbon dioxide and water. Fermentation, on the other hand, results in a net gain of 2 ATP molecules from the partial breakdown of the glucose molecule.